Dear students, in this session, we are going to discuss bacterial growth curve and its significance. How do we get or how do we plot a bacterial growth curve? When a fresh nutrient medium is inoculated with a given number of bacterial cells and the population growth is monitored over a period of time, then we can plot the bacterial population against the time of incubation. Plotting the data by putting the number of bacterial cells along y-axis and time along with x-axis will yield a bacterial growth curve. A typical bacterial growth curve have these four distinct phases: lag phase, logarithmic or log phase, stationary phase, and decline or death phase. So, this is a typical bacterial growth curve. There are four different phases: lag phase, log phase, stationary phase, and death or decline phase. In the first phase, called the lag phase, the population remains at the same number as during this phase the bacteria are becoming accustomed to their new environment. So during this phase, metabolic activity is taking place in the bacterial cells and some repair and the production of new materials within the cells are being taken place. So this is the lag phase. The next phase is the logarithmic or exponential phase. During this phase, the bacterial growth occurs at its optimum level and the population doubles rapidly. This phase is represented by a straight line and the population is at its metabolic peak during this particular phase, during log phase. The next phase is the stationary phase, and during this phase, the reproduction of bacterial cells is offset by their death. So, the population reaches a plateau. The reasons of bacterial death during stationary phase includes the accumulation of metabolic waste, the lack of nutrients, and the unfavorable environmental conditions that may have developed. And after stationary phase comes the death or the decline phase. If the conditions, that is the metabolic waste accumulation, the lack of nutrients, etc., if these conditions are not altered, then the population will enter the decline or the death phase. During this phase, the bacteria will die off rapidly and the curve turns downwards, and soon the population reaches its decline. So, in a growth curve, these are the four distinct phases: lag phase, log phase, stationary phase, and death phase. And we get a growth curve when we plot the time of incubation versus the number of cells or the population size. So, now we will discuss the four phases in detail. The first one is the lag phase. This phase is also known as the preparatory phase or the phase of adjustment. So, this lag phase is the first phase in a growth curve when microorganisms are introduced into fresh culture medium. Usually, there is no immediate increase in the cell number, and therefore this period is known as the lag phase. There is a lag in the increase in population size. This is the period of time required for the inoculated bacteria to get adjusted to the new environment. They have to get adjusted to the changes in temperature, pH, nutrients, etc., which they are going to face in this particular new cultural conditions. Although cell division does not take place immediately, and there is no net increase in mass, that doesn't mean the bacteria are not having any metabolic activity. They are actively metabolizing and synthesizing new cellular components during lag phase. The length of lag phase varies in length considerably depending upon the condition of the microorganism and also depending upon the nature of the culture medium. So what is the need? What is the necessity for a lag phase? It is needed for a variety of reasons. The cells which we are going to inoculate or which we inoculated into the media, they may be old and they may be depleted of ATP, essential cofactors, ribosome, etc. All these will be synthesized during the lag phase. The second reason, the media may be different from the one in which the microorganism or the bacteria was growing in previously. So new enzymes would have to be synthesized so that the microorganisms or the bacterium will be able to use the nutrients that are present in the present medium. And there are chances that the microorganisms in the inoculum have been injured. They may have damages in their cell wall or membrane, plasma membrane. So all these damages, all these injuries have to be repaired. So the repair of these injuries or damages will take place during lag phase. The length of the lag phase also depends upon the size of the inoculum. If the inoculum contains high number of cells, then the lag phase will be smaller. And vice versa, if the inoculum contains very small number of microbial cells, then they will need a more lengthier lag phase. Also, if the inoculum is from an old culture or a refrigerated culture, and if it is inoculated into a chemically different medium, then the lag phase will be quite long. Similarly, the lag phase will be shorter if a young, vigorously growing culture is transferred to a fresh medium of the same composition. Then the cells are not having any damages. They are not having any requirement, any additional requirement since the medium is of the same composition. So they will get adjusted to the new conditions rapidly, and as a result, the lag phase will be minimum. So during the lag phase, the cells retool, they begin to increase in their size, their mass, they will replicate their DNA, and they finally start to divide. Now we will discuss the characteristics of a lag phase. During lag phase, the metabolic activity of the cells increases. The cells starts synthesizing various proteins, RNA, etc. And there will be increase in the volume or mass of each cell. The cells will repair their damaged parts. There is no appreciable multiplication of bacteria occurring during lag phase. There is no increase in the bacterial population or there is no increase in the number of bacterial cells during a lag phase. Generally, depending upon the type of microorganisms and also on the cultural conditions, the required time duration for a lag phase varies between 1 to 4 hours. Now, the importance of lag phase. During lag phase, the organisms will be susceptible to certain antibiotics, membrane acting antibiotics such as polymyxin, amphotericin, etc. Or they are also sensitive to detergents, soaps and other surface acting agents. The next or the second phase in a bacterial growth curve is log phase, which is also known as exponential or logarithmic phase. So during the lag phase, the bacteria are getting accustomed or adapted to the new environment, and once their metabolic machinery is running properly, the bacteria starts dividing by binary fission, and as a result, they will double their number. This is an exponential or log phase, and during this phase is the maximum growth rate of the population achieved. The microorganisms are growing and dividing at the maximum growth rate possible. And the maximum growth rate depends upon their genetic potential, nature of medium, and also on the conditions or the growth conditions in which they are growing. As we said, the microbial cells are dividing at their maximum growth rate possible, and the rate of growth at which the bacterial cells are dividing is constant during the exponential phase. They are dividing at regular intervals. This phase is termed as exponential phase since rapid multiplication and increase of cell numbers occurs geometrically or exponentially. And during this phase, the microbial population is most uniform in terms of chemical and physiological properties, and thus the cultures during their exponential phase are usually used for biochemical and physiological study purposes. Now let us discuss the characteristics of a logarithmic phase, of a log phase. So we said during log phase, exponential or geometrical increase in population size is occurring. There will be rapid multiplication and increase of cell numbers which occur in a geometric or exponential manner. So, with each generation, the cells are getting multiplied in their number. The number of cells getting doubled during each division of each round of multiplication. And during log phase, the metabolic activity is very high. There will be active synthesis of cell wall. Generally, the time which is required for an exponential phase varies from 1 to 4 hours, and it depends on the availability of nutrients. Now, what is the importance of log phase? During log phase, the microbial cells are more susceptible to antibiotics that inhibits the cell wall synthesis of protein, DNA replication, etc. During this phase, the virulence or the disease causing capability of bacteria such as pathogenic bacteria will be the highest. We already said that during exponential phase, each microorganism is dividing at constant intervals, and this particular interval which is required for a microbial cell to divide is known as the generation time. So it is a time required for a cell to divide. Similarly, it is a time required for a microbial population to double in its size. So this is the generation time or doubling time. So in other words, generation time or doubling time is a time required for a bacterial cell to divide into two, or it is a time required for a bacterial population to double in its size. If a culture tube, a culture medium is inoculated with one single cell that divides every 20 minutes, if the generation time is 20 minutes, then the population will be 2 cells after 20 minutes, 4 cells after 40 minutes, 8 cells after 80 minutes, and so forth. Because the population is doubling during every generation, the increase in population is always 2 raised to n, where n is the number of generations. The resulting population increase is exponential or logarithmic. So, if n0 is the initial population size, and t is the population size at time t, and small case n, it is the number of generations at the time t, then nt is equal to n0 multiplied by 2 raised to n. If we apply logarithm, on both sides. Log nt is equal to log n0 added with n log 2. We can calculate the number of generations n from this equation, which is equal to log nt minus log n0 divided by log 2, which is equal to, if log 2 could be rewritten as 0.3010, then the equation comes like this: log nt minus log n0 divided by 0.3010. This is the number of generations at time t. The rate of growth during the exponential phase in a batch culture. The rate of growth during exponential phase can be expressed in terms of mean growth rate constant, which is written as K. This is the number of generations per unit time. Number of generations per unit time is the mean growth rate constant. So K is equal to n by t, which could be rewritten as log nt minus log n0 divided by 0.3010. This is n divided by t. So it is log nt minus log n0 divided by 0.3010 t. This is K, that is the mean growth rate constant. Now we will discuss the mean generation time or mean doubling time, which is denoted as G. If the population doubles at a time t by taking a time t, then this time period t will be equal to G, that is the generation time or the mean doubling time. Then we can consider the population at a time period of t is equal to 2n0, that is double the number of initial population size. So if a population doubles, then the population size nt is equal to 2 multiplied with n0. So under this situation, let us see what the mean growth rate constant K will be. K is equal to log nt minus log n0 divided by 0.3010 t. We can replace nt with 2n0, and since t is equal to g under this specific condition, if the population is getting doubled at a period of time t, then we can replace t with g. So the k will become like log multiplied by 2n0 minus log n0 divided by 0.3010 g. We can rewrite this equation like this: log 2 plus log n0 minus log n0 divided by 0.3010 g. Log 2 is equal to 0.3010. So this equation could be rewritten like this: 0.3010 plus log n0 minus log n0 divided by 0.3010 g. So in this equation, the 0.3010s will cancel out each other. Log n0, it will also cancel out each other. So the equation becomes K is equal to 1 by G. That is, the mean generation time or the mean doubling time G is inversely proportional to the mean growth rate constant K. G is equal to 1 by K, or K is equal to 1 by G. Mean generation time is reciprocal of mean growth rate constant. The mean generation time or G can also be determined directly from a semi-logarithmic plot of growth curve. We can calculate generation time by extrapolating the population size to time. If we have plotted a growth curve with time on x-axis and number of cells on y-axis, by extrapolating the population size to the time, we can get the generation time. We can calculate the generation time from the growth curve, from the semi-logarithmic plot of growth curve. So here, if we extrapolate from the number of cells from y-axis to the x-axis, we get 2 points, and the difference between these 2 points gives the generation time. So, the mean generation time can be determined directly from a semi-logarithmic plot of the growth curve. And if we have the g value, then we can calculate the k value or the mean growth rate constant, since they are inverse to each other, reciprocal to each other. The calculation of generation time from a growth curve is done by extrapolating the population size, which is given in the y-axis, to the time, which is given in the x-axis. The generation time varies markedly with the species of microorganism and also depending upon the environmental conditions. It ranges from less than 10 minutes for a few bacteria to several days with some other microorganisms. The generation time in natural environment is usually much longer than that in a laboratory culture. So this table gives the generation time of different bacteria at different different conditions, temperature conditions in which they are being incubated. So we can see that certain microorganisms, certain bacteria like Bacillus stereothermophilus have a very short generation time, while some other microorganisms like Mycobacterium, Treponema, etc., they have very long generation times. Coming to stationary phase, in this phase, which is attained by bacterial culture at a population level of 10 raised to 9 cells per ml, the total number of viable microorganisms remain constant. This is due to the balance which is attained between the rate of cell division and the rate of cell death. The growth rate of the bacteria decreases and the number of bacteria stabilizes. So, during stationary phase, the total number of viable microorganisms remain constant due to the balance between cell division and cell death. During stationary phase, the bacterial cells will become more and more resistant. They respond to the changes during stationary phase by certain morphological changes such as endospore formation. Certain bacteria will produce endospores. There will be increased peptidoglycan cross-linking in their bacterial cell wall. Some bacteria produce starvation proteins, and DNA binding proteins will be formed which will protect the DNA, and to protect proteins from the adverse conditions during the stationary phase, certain molecules such as chaperones will be produced. As a result of these morphological, biochemical and physiological changes, the bacterial cells will become more and more resistant to the conditions such as starvation, temperature changes, oxidative and osmotic damages, and the presence of toxic chemicals, etc., which are prevailing during the stationary phase. So, why the microbial populations reach this particular stationary phase where their growth rate decreases, their number of viable cells will become constant, and all the cells which remain viable during the stationary phase remain more and more resistant. What is the reason by which the microbial populations reach this stationary phase? The first and the prime factor is the nutrient limitation. Due to their continued growth, the microbial populations will be using the nutrients which are available in their culture medium, and as a result, the essential nutrients will be getting depleted, and as a result, the population growth will slow down. Another important factor is the oxygen availability. The growth of aerobic microorganisms are limited based on the level of dissolved oxygen available. Since oxygen is very poorly soluble, only the surface of the culture media will have adequate oxygen concentration to support the microbial growth, especially the growth of aerobic microorganisms. The cells which grow beneath the surface towards the bottom or middle areas of the culture medium, they will not be able to grow unless the culture medium is properly shaken or aerated. So oxygen availability, the decrease in the availability of oxygen is another factor which influences the microbial growth. The next factor is space limitation. Due to their increased growth during the logarithmic phase, the population size will be increased, and we said already that the stationary phase is generally attained in a bacterial culture when the population level reaches about 10 raised to 9 cells per ml. So there are increase in the population size which will result in space constraints. There will be limitation of the biological space available for growing bacteria. The next factor by which the microbial population reach stationary phase is the accumulation of metabolic end products. During their growth, the microbial cells, the bacterial cells will be producing certain metabolic end products as a part of their metabolism, and this accumulation of these toxic waste products will result in a decrease in the population growth, will result in the decrease of microbial growth. For example, certain bacteria, they will produce organic acids during their growth, during their fermentation, and as a result, along with their growth, the medium, the culture medium in which they are growing will become more and more acidic. And as a result, the bacterial cells will not be able to grow further. Since due to the acidic pH, their growth will be inhibited. Now, what are the characteristics of stationary phase? There is no net increase in cell number. Due to the balance between cell division and cell death, there is no net increase in the cell number. The size of the population remains constant. The rate of cell division is balanced with the rate of cell death. And generally, the time required for attaining a stationary phase depends upon the type of the organism and also on the cultural conditions, but generally it ranges from few hours to few days. And during stationary phase, some bacteria start to produce exotoxins. Some bacteria produce endospores, and some bacteria start to have erosion in their peptidoglycan layer, and mainly in gram-positive bacteria, the erosion of peptidoglycan layer in such gram-positive bacteria will result in showing such cells as gram-negative upon staining. Due to their damage in their peptidoglycan layer, such gram-positive cells will appear as false gram-negative upon staining. So during stationary phase, some bacteria release exotoxin, some produce endospores, and in some gram-positive bacteria, there will be erosion of peptidoglycan layer. The next phase is decline phase or death phase or lysis phase. In this phase, the total number of viable cells decrease in a rapid rate. And this decline or this decrease in the microbial population is in a logarithmic pattern. That is, a constant proportion of cells die every hour. And this decline in microbial population is due to the detrimental environmental changes that are happening in the culture medium, such as there will be nutrient deprivation, buildup of toxic waste, etc. And as a result, the viable cells will not grow, will not multiply, and many of them start to die, and as a result, the number of viable cells will decrease rapidly. The characteristics of a decline phase is that the death rate is higher, greater than the growth rate during a decline phase. And it is due to this factor is that the microbial population is getting reduced rapidly. The death rate is greater than the growth rate. And there will be accumulation of higher amounts of toxic metabolites. During stationary phase itself, there were accumulation of toxic metabolites. The metabolites which were produced during the normal metabolism of bacteria, such toxic metabolites will be accumulating in higher and higher levels during decline phase. And several bacteria, sporulating bacteria, will produce their endospores during decline phase, and some bacteria start to produce and release their endotoxins during decline phase. So, we were discussing about the growth curve, its various stages. Thank you so much for listening.